we have very few limited staff. And if you have these experts, we could have them in oncology, we could have them in cardiovascular drugs, pediatric drugs, etc. And they will be able to review some of the clinical trial reports and advise the agency accordingly. The agency has also put in place the relevant clinical trial guidance documents, such as the Good Clinical Practice Guidelines, the GCP inspection checklist, clinical trial application format, serious adverse events during clinical trial reporting format and form, guidance document for collection and exportation of clinical trial samples obtained from trial participants, clinical trial documentation guidelines, etc. Please serve as a guide to the intended clinical trial investigator or sponsor. Prior to the workshop, these documents were not placed. But since that period, we worked hard to make sure that these documents, which we've been toying about, that now everything is reviewed and are in place to guide us to ensure that we have a robust regulatory framework for clinical trials in Nigeria. All these efforts are geared towards ensuring that all trials to be conducted in the country are done in accordance with ICH Good Clinical Practice Guidelines, the Helsinki Declaration, as well as to ensure that all products to be registered and used in Nigeria are safe and efficacious. It is necessary to note that most applications received in Nigeria are phase 2 and phase 3. So that's a typographical error. It's phase 3, not phase 4. Most clinical trials we receive are phase 2, very few phase 2 and phase 3. What does this tell us? Basically, research is not going on in the country. And most of them are from outside the country. In the morning, Dr. Yetunde Taiwo told us about the problem, the constraints, infrastructure. The present position does not encourage research, and that's why we cannot have phase 1, phase 2. We do not have people coming to us pre-submission application for us to discuss and dialogue about the research going on before they get to phase 3. We don't have such cases because there's no research going on. By the time we get applications, it's mainly phase 3, and sometimes phase 4 when everything is. Actually, for that phase 4, we demand for it to give us a documented plan for phase 4. Especially for products that, well, they have done the studies in other black community or black population, and yet we still want to have some data generated within our own locality. And that's when we ask for this phase 4. So far, we have only a total of 34 clinical trial applications that have been approved, very small limited, consisting of 28 conventional drugs, 3 herbal and 3 biologicals. Next slide. Well, we don't charge much. We are contemplating increasing the charges, but all the same, we left these charges to encourage people to come, so that nobody will say that because the charges are so much, that's why they cannot come. But I'll use this opportunity to also extend our appreciation to GSK. GSK builds a clinical trial site in Nigeria, in Lagos, which is very good, which is encouraging. And they are using it to carry out clinical trials in Nigeria. Challenges. Most applicants, that is sponsors and investigators, lack basic GCP knowledge. That is the truth. Because since they are not involved in research, they are lacking in the basic knowledge. Because if they are into research, if the infrastructure is there, then it will be easy for them and for us too, that are the regulators. Then on our side, we don't have enough staff. And then, not only not having enough staff, skill to review new innovations like the biopharmaceuticals and medical devices. I had the opportunity in May to visit the US APA laboratories here for medical devices. I was amazed. We are far. I don't know when we get to that level. Even China, that is an emerging economy, in China, I visited many laboratories there. Only one laboratory is developing a laboratory that can analyze the influence of radio frequency on medical devices. It's unfortunate. And there's actually, it's from Germany. That's to tell you the level of technology, that emerging technology, and we are lacking behind. When we don't have the basic infrastructure, how do you talk about high-level technology? Lack of awareness among healthcare professionals about NAPPSA's role in regulating clinical trials. We understand that some clinical trials are ongoing in the country without NAPPSA or Federal Ministry of Health or NREC knowing about it. It's either the people they are not aware, and we believe they are not aware. And that's why we are making efforts to now do enlightenment campaign and to make sure we organize seminars for all stakeholders. Then the infrastructure, I've spoken about that. The agency is making effort to tackle these challenges by training and retraining her staff. The agency is also planning to have a forum for all stakeholders. These stakeholders, that is the industry, because it would be fruitless if the regulatory authority builds capacity, has a robust regulatory framework for regulating clinical trials in Nigeria, while the industry is not taking and is not trained. They are not aware. They don't know the demands or the requirements for good clinical practice. So for that, we are trying to organize a workshop where the industry will be brought in, all stakeholders, people in government, so that they will be aware of the requirement and the need for that infrastructure to have things that will encourage clinical trials in Nigeria, and then so that Nigeria can be a destination for clinical trials. Because we know what the other countries are benefiting: India, China, South Korea. Finally, I wish to call on all stakeholders in the industry to partner with NAPPSA to make Nigeria a destination for clinical trials in Africa. This is achievable because of the expertise that has been developed over time within NAPPSA, coupled with the country's large population of qualified personnel, both within the country and in the diaspora. I urge you once again to avail yourselves the opportunity to contribute to the building of a strong and vibrant clinical trial industry in the country. Thank you very much.
Where we are with clinical trials regulation in Nigeria, I would say that we should hold off on questions to the end of both presentations, because some of the questions may be relevant to both speakers, and we can have an integrated type of response. The next presenter is Dr. April Brady. She's currently a team leader in the Division of Bioequivalence 1 in the Office of Generic Drugs, CEDA, at the US FDA. Dr. Brady?
Drug Administration. The outline of my presentation, first I will be giving you a historical perspective of the US FDA, and then I will be giving definition of some key terms that are used by the agency to define generic drug products. Then I'm going to discuss the description of the route of drug administration and dosage forms. Thirdly, I'll be talking about the current federal regulations and guidances for these particular drug products. Then I'll go into talking about the current scientific methods and tools which we use to approve these products. And lastly, I'll give an overview of the different scientific methods and tools that we're currently reviewing and exploring for these particular products. Okay, historical perspective. In 1931, the US FDA was formed. Prior to 1931, there was no regulatory body that was governing food and or drug products. So basically, drug companies could have a drug on the market with no pre-clearance from the government. In 1938, we had the passage of the Federal Food, Drug, and Cosmetic Act by the U.S. Congress. And what this did was it gave the U.S. government the ability to not only regulate food and drugs, but also cosmetics and therapeutic devices. And one of the key components of this act was that it now required drug companies to show that their products were safe. So the drug companies now had to conduct clinical trials. Prior to 1938, they did not have to do that. In 1962, we had the passage of the Keeper-Harris drug amendments, which now required for drug applicants to show that their products were effective as well. And then in 1984, we had the passage of what is known as the Drug Price Competition and Patent Restoration Act, which is known as the Waxman-Patch Act by the U.S. Congress. And this allowed for the approval of generic drug products through abbreviated new drug applications in which a product can now demonstrate bioequivalence to the innovative drug product. Some of the benefits for the innovator was that it restored the effective patent terms for 20 years. It allowed for up to 5 years additional patent protection for the new medicines developed to make up for time lost while they were undergoing approval. And lastly, gave a 30-month stay procedure to allow the patent holder the opportunity to obtain a court ruling as to whether or not a generic infringed upon its patent. But overall, the goal of this act was to make available low-costing, safe, and effective drug products to the American public, which it has accomplished. Okay, now I'm going to transition into talking about key terms in the description of the route of administration and dosage forms. According to Title 21, Code of Federal Regulations, Part 320.1, bioavailability means the rate and extent to which the active ingredient or active moiety is absorbed from a drug product and becomes available to the site of action. Topical drug products are locally acting. Therefore, bioavailability may be assessed by measurements intended to reflect the rate and extent to which the active ingredients or active moieties become available to the site of action and that are not absorbed. Also, another term that we commonly use within the agency is bioequivalence, which means the absence of a significant difference in the rate and extent to which the active ingredient or active moiety in pharmaceutical equivalents or pharmaceutical alternatives becomes available at the site of drug action when it's administered at the same molar dose under similar conditions in an appropriately designed study. So for generic products, what you're trying to show is you're comparing a test and a reference product, and you want to show that there's an absence of a significant difference between the products. Pharmaceutical equivalence simply means drug products that are identical in dosage forms, identical in the amounts of the active drug ingredient. So when I'm using that term, that's what the agency defines as a pharmaceutical equivalent product. According to our Orange Book, and this is available to the public, is a listing of all approved drug products with therapeutic equivalent evaluations. Products that are defined to be therapeutic equivalents means drug products that are considered to be therapeutic equivalents only if they are pharmaceutical equivalents and if they have the same clinical effect and safety profile when administered to patients under the conditions as specified in the labeling of that product. Okay. Now I'm going to transition into talking about the route of administration. Most topical drug products are administered to the skin or become available to the skin as the site of action. And on the left-hand side, you can see the schematic of the structure of the skin: striatum corners, top layer, epidermis, dermis, and fatty tissue with your blood vessels. The skin accounts for more than 10% of the body mass, and it's in direct contact with the environment. Some of the major functions is that it's protective, it maintains homeostasis, and it's sensitization, and that you feel heat, pressure, pain, and allergens. Next slide. Some of the sites of action for topical dosage forms is the face for acne, hands, your arms, your legs, back, your scalp. Next slide. According to the U.S. Pharmacopoeia, there are different categories for topical dosage forms. You have your solutions, your semi-solids, and your solids. The solutions are commonly emulsion solutions and suspensions, and semi-solids are like creams, foams, gels, ointments, paste like toothpaste, and some of the solids are powders and transdermals. The products that are marked by X, like the little red X, denotes dosage forms that I will not discuss any further in this presentation. So what I'm discussing as far as the regulations and the guidances do not refer to these products. Okay. This is just a schematic showing the dosage form performance when you're comparing, like say, oral drug product which is absorbed versus a topical product which is made available to the site of action. If you look at on the oral side, you have the drug that's released into solution and is absorbed through the gut wall. It goes into systemic circulation and then to the site of activity and produces its therapeutic effect. Whereas you have a topical dosage form in local action, where you see the number 1 is designated skin path 1. It becomes available to the site of action and site of activity, and then it produces its therapeutic effect. And then you may have some of the drug that actually goes into systemic circulation, but in most cases it may be negligible. There are other paths as well. If you designate by number 2, where some topical products go directly into systemic circulation. And then there's also a possibility that there's another skin path which is different from skin path 1, which goes to the site of activity and produces a therapeutic effect and goes to systemic circulation. Okay. Next slide. Okay, now I'm going to talk about the current federal regulations and guidances for topical drug products. And the difference between regulations and guidances is that regulations are law, means that it governs not only the agency but also the applicants. Guidances are simple recommendations to applicants as to how they should proceed in conducting studies for these particular products, and also how the agency reviewers should review these particular applications when they come into the agency. Currently, the agency has a website which is available to the public, which gives a listing of, it's not for all generic products, but it has a listing of a lot of generic products. So if you want to know as to what studies should be conducted for products, you can go to the FDA website and it gives a listing for that. It tells you what the reference product is, what studies should be conducted, if there are any safety issues. It goes through all of that and provides you with all that information. Okay, regulations for topical products. For topical products that are applied to the skin and are in a solution form, they can be granted waivers. And basically, a waiver is where the firms do not have to conduct any type of bioavailability or bioequivalence studies in order to show that their products are bioequivalent to the reference product. And there are 3 caveats for this. First off, it has to contain an active ingredient in the same concentration and dosage form as a fully approved new drug application or abbreviated new drug application. And secondly, it cannot contain any inactive ingredient or other change in the formulation that may, formulation that is subject of the full new drug application or abbreviated new drug application that may significantly affect absorption. And for the case of locally active drugs such as topicals, they cannot significantly affect systemic or local availability of that particular drug product. If they meet these criteria, then the agency can and will grant a waiver for these products. Okay, this is just a listing of some of the examples of topical solutions in which we granted waivers, cyclopyrox, Finlac, minoxidil, Rogaine for hair growth, fluoxamide, Lidex. So it's just a listing. Next slide. Also, federal regulations, Part 3, 2024 gives the detail, the types of evidence in which we measure, which are used to measure bioavailability or establish bioequivalence for drug products. And this schematic is just showing you in descending order what the agency prefers. So normally we'll recommend in vivo pharmacokinetic studies. If that's not feasible for a product, then we'll recommend in vivo pharmacodynamic studies. If not, clinical bioequivalence trials with clinical endpoints. If not, in vitro test approach. If not, any other approach deemed acceptable. I'm going to talk specifically to each one of these points as it pertains to topical products in part 4 of my presentation. And now I'm going to transition into the guidances. We have 2 guidances in place that are specifically for topical products. The first one is the guidance for topical dermatological corticosteroid products, and this is specifically for corticosteroid products. And in this guidance, we recommend vasoconstrictor assay studies, which have PD endpoints, pharmacodynamic endpoints, which is known as skin blanching. I'll talk a little bit more about this as well in section 4 of my presentation, in the next section of my presentation. And this is available publicly as well on the agency's website. The next guidance is for non-steroid semi-solid dosage forms, which are like gels, lotions, creams, these particular types of products. And it's the scale-up and post-approval changes, chemistry, manufacturing, and controls, in vitro release testing, and in vivo bioequivalence documentation. And we want to make some type of modification to their product, whether it be in the manufacturing of the product or be in the formulation of the product. And this is guidance as to how they go about into getting approval for those changes to still show that their products are bioequivalent to the originally approved drug product studies for topical drug products. And again, this is just a schematic, just a different layout of the schematic that I showed earlier, where you see the topical dosage form performance in general skin path 1, and the site of action is available to the site of action. So you're going to see your therapeutic effect first. Then you may see a negligible amount in your systemic circulation. You think about the structure of the skin, it has to go through several layers of the skin before it gets to the blood vessels to be absorbed into the bloodstream. Okay. In vivo pharmacodynamic studies. The pharmacodynamic studies contain an endpoint which is indicative of drug delivery to the site of action, which is the skin. For dermatological corticosteroids, the PD endpoint is skin blanching, which is the lightening of the skin. And basically what happens is the subject's arms, there's a certain amount of drug that's applied. I'm going to really see it in this slide. There's a certain amount of drug applied to a certain area. And over time, the lightening of the skin is measured. And then based on the results obtained from that, statistical analysis is conducted on that data. Next slide. Okay, this is just going into more detail concerning the guidance for dermatologic corticosteroids, in which we recommend vasoconstrictor assay studies. The pilot dose response study is a dose duration response relationship for drugs in the term of the EMAX model, which is the pharmacodynamic model, and I'll show you on the next slide the mathematical equation which represents that, and it's conducted on the reference product only. And in this dose duration response study, you actually get the ED50, which is the half maximum effective dose in unit time, is actually measured in this study and is used in the pivotal study. And normally about 20 to 30 healthy subjects are enrolled in these particular studies. Then you have the pivotal bioequivalence study, which is the comparison of your proposed generic to the reference drug product. And there are 3 doses. You have D1, which is your shortest duration, which is half of the ED50. Your D2, which is your longest duration, which is 2 times your ED50. And on the next slide, there's actually a schematic which will show you the effect dose curve, so you get an idea of why these 3 points are critical, why these 3 dosage points are critical for the study. And normally about 40 or more healthy subjects are enrolled. However, the potency of the drug product is the determining factor as to how many subjects are enrolled in the study. So if you have a topical corticosteroid with low potency, you're going to tend to need more subjects enrolled in the study versus a topical product that has high potency. Subjects are then screened as PD responders with a D2 greater than D1 ratio that meets the specified minimum value and subsequently may be included in the data and statistical analyses supporting bioequivalence. And our 90% confidence interval normally is 80 to 125%. Okay, in the next slide, we look at the bottom, the effect versus dose curve. The bottom star represents D1, and this is when you're first starting to see a pharmacodynamic effect. And then the top star is D2, where you're starting to see the effect right before it plateaus, and then in the middle you have your ED50. So this is the window in which you want your dosage times to be measured. So you're capturing what is actually going on with the product. And then on the left-hand side, you have the pharmacodynamic Emacs model, which is just showing you mathematical representation of the pharmacodynamic effect model, which is the same as the Emacs model for dermatologic products data. It's just that the units that are used to express things are a little bit different, but it's the same thing. Next slide. And then here is an example of some of the topical corticosteroid drug products. Clobetasol propionate is classified as a class 1, which is very high potency, whereas you have dioxamethasone, which is classified class 2 as high potency, and you have betamethasone, which is medium potency, class 3, and then you have aclamidazole, which is a class 4, low potency. So go down the list, a higher number of subjects that would need to be enrolled in this study to show bioequivalence to the reference drug product. Okay, bioequivalence trials with clinical endpoints. In general, we tend not to try to recommend these just because they are costly and they require enrollment of a lot of patients in these particular studies. However, the clinical endpoint is a measurable clinical parameter of treatment outcome that can be used to ensure equivalent delivery of the drug to the site or sites of action by showing the same effect. And some of the types of products that we recommend this for are antifungals, acne treatments, and topical shampoos. And the types of endpoints are dichotomous endpoints where you have a cure or success versus failure, like at an athlete's foot, or continuous endpoints where you have a change from baseline in lesion counts or symptom scores, like inflammatory, non-inflammatory lesion counts for acne or papular-pustule counts for rosacea. We have an Office of Medical Affairs in the Office of Generic Drugs, which actually handles the reviewing of these particular applications, these particular submissions, and Dr. Lena Hickson heads that group. Okay, now I'm going to talk about the in vitro studies, the SUPAC guidance, which deals with the semi-solids, the creams, gels, lotions, and ointments. And the in vitro surrogate test may be used under certain conditions, certain changes, to ensure that product quality and performance shows the sameness are maintained over time in the presence of change, pre and post change. This is applicable to both new drug applications and abbreviated new drug applications. And it allows for changes in components or composition, manufacturing, processing, and equipment, scale up, scale down, or manufacturing, or changing in the site of manufacture of the product. Based on, there are 3 level changes, and based on those level changes depends on what the agency recommends as far as what the firm needs to do in order to show sameness between the products. Sometimes if your plant you're changing from one plant to another plant that's only 5 miles apart, minimal information is going to be required from you. Whereas if you're completely changing, you have 6 excipients, you're changing 3 out of those 6 excipients, more than likely you're going to have to conduct some type of additional bioavailability and a bioequivalence study. Whereas if in vitro studies, if there's some like one of your excipients you're changing maybe within 5 to 10%, then we may ask for just in vitro studies, as long as that particular excipient does not affect the availability of the active ingredient, then we may just request an in vitro test. So it really depends on the changes that are being made to the product. Next slide. Some caveats to this is that the development and validation of an in vitro release test are not required for approval of a new drug application or an abbreviated new drug application. Nor is the in vitro release test required as a routine batch quality control test. Also, the in vitro testing alone is not a surrogate test for in vivo bioavailability or bioequivalence. And lastly, the in vitro test should not be used for comparing different formulations across manufacturers. And this is just a slide showing you, in our guidance, we actually recommend, so if the firm decides to use a different method to show, a different in vitro method to show equivalence between their products, then the agency can review that, but currently we recommend the France cell system. And this is just where you look at the schematic, the test product is placed on the upper side of the membrane in the open donor chamber of the diffusion cell, and the sampling fluid is placed on the other side of the membrane in the receptor cell. And then the guidance goes into more detail as discussing the in vitro study design as far as the diffusion cell system, the membrane, receptor cell, number of samples, and what type of statistical procedure is used to show bioequivalence. Next slide. Okay. Last thing we're going to talk about what methods that are currently being explored and are developed for these particular products. In 2007, the agency implemented what is known as critical path initiatives. And these critical path initiatives included generic products. And in section 4.33, it focused on bioequivalence of topical dermatological products. And the three key areas was the design of bioequivalence trials with clinical endpoints. And this is in the selection of more relative endpoints that require less patients to be enrolled in the study. And also in vitro characterization of topical dermatological products whose formulations are identical to each other, and using rheological test methods and diffusion cells. And then lastly was the local delivery of topical dermatological products. And this area covered possibly looking at pharmacokinetic studies for some topical drug products. Skin stripping technique has been discussed for years with the agency. And so they're revisiting that as well. And then microdialysis and near-infrared spectroscopy. And that's it. These are just some references for the information that I provided in my presentation. And these are my acknowledgments. And that's it.
Well, thank you, Dr. Brady, for that very comprehensive presentation on requirements for clinical development of dermatological products. Well, I guess, we have a limited panel today because some panelists were not able to be here today. So we can now go into, the questions and answers session. Please, I would ask that you be as precise as you can about the questions you ask. Thank you.
My question is for Dr. Maduke. Thank you very much for the lovely presentation. My first comment is actually not a question. I'm pleased to know that NAPPSA has a clinical trial doing now. Even I didn't know it until today. So, if there are many people who are conducting trials in Nigeria without contacting the office, it's probably because they didn't know, which is unfortunate. And I think, and it should be illegal actually. So I think it's more than just spreading the word that you exist, but making sure that anyone who is conducting it has to feel some pain for conducting it without contacting NAPPSA. But the main thing, a great deal of enlightenment will help. Now the question I have is, you indicated in one of your slides that you do have phase 4 studies required of anyone wanting to register drug candidates in Nigeria. Can you give me an example of some examples of what you expect to see in a phase 4 trial? And I'll let you know where I'm coming from right away. Maybe you can address that as well. Because the best way I can say it, and it's something that I've thought about for a very long time, is Nigeria, a country like Nigeria doesn't have a lot of resources to go through the regulatory requirements of, that an FDA here would go through to approve a drug. And I used to feel quite offended, I must say, when I worked for the agency and we were putting together the ICH harmonization between Europe and the United States. And Japan was part of the group. And WHO represented Nigeria and the rest of the world. As because there was no such regulatory mechanism. And my concern was, how can, there are racial issues related to drugs going across borders and across continents. And there have been deaths or a serious adverse effects that were not seen in prior trials. So I'll give a quick example and then the question. This morning I gave a talk on biomarkers for discovering or identifying responses or lack of responses to certain drugs. There is an anti-cancer drug called irinotecan, and there is a marker for it. When the drug got on the market, it got to Japan, and there were deaths in Japan that were not seen in the original trial. And it turned out that the Japanese had a mutation to the drug that to the gene that converts that drug from the prodrug to the active drug, SN product. Now, what I'm trying to say is, do we, in these phase 4 trials that we do in Nigeria, conduct bridging studies? That's really the study that's required for any new drug that's entering a market like Nigeria or Japan. It's a small study that shows that that drug is safe in that population of candidates. A G6PD is another gene. Do you want me to paraphrase the question?
No, answer. You are still talking.
I'm through now.
Okay, thank you very much. Well, sorry, that slide, remember I said that 4 is 3, it's a typographical error. Then I went on to explain that we ask for a type of plan that is post-marketing surveillance, sort of. When a product, when a study has been carried out in some countries, and then maybe there are some areas, gray areas, which we really want to really have more evidence on, we can now say, give us a planned phase 4. We now call it planned phase 4. But not usually what you normally have in our clinical trial submissions are mainly phase 3. Now this phase 4 is those ones, phase 4, you have a very large population. The drug for phase 4 has got marketing authorization. But normally, when we have such things, we ask them to give us a planned phase such that we follow it up very well ourselves okay, we will now know the hospitals and where the whole thing should be going so that we now monitor very well, especially where you have some gray areas to, to see, which we are not comfortable with in the studies they have conducted in some other black population. That's where we have such plan phase 4.
Okay, so the phase 4 that you have conducted is really similar to post-marketing studies
Yes.
here.
Yes.
Okay.
Yes.
What I was actually asking about also deals with very specific and required bridging study.
Bridging studies.
Yeah.
Yes, we also have the bridging studies, whereby, for example, somebody can say, yes, I conducted a trial in US here, and then there were some black people that participated, and then we look at it, and the population of the black people that participated is not, does not represent the percentage that we would be very comfortable with. Then we have such big studies. I'm using that as an example. Sometimes we have big studies.
So you make them do it in Nigeria?
Yes.
Okay, good. Thank you.
Thank you.
Can you just please identify yourself briefly before the question?
Okay, I'll do myself. Thank you. I am Ernest Izeli. I'm professor of biology and biochemistry at Jackson State University in Jackson, Mississippi. I came from Nigeria for this convention because I'm in Nigeria doing a sabbatical, 6-month sabbatical. My question to you, ma'am, is do you have a duration requirement? How long should a clinical trial be? Or does it vary from study to study? And do you also have participant requirement in terms of the number of individuals that you consider adequate? I'm not sure if that one is included in your guideline. And finally, beside the fee of $50,000 that you charge, do you provide other logistical support to in other words, does the Nigerian government provide any funding for some of this trial? That is, or you're one of that. If you're the one doing the studies, you pay for it. How does that work? Thank you.
Thank you. Well, we cannot determine the period for you to conclude your studies. Yes, because you design the protocol, we look at the protocol to ensure that all the relevant aspects have been covered in the protocol. And basically, remember, it's for the protection of the subject, the human subject that is participating in the trial. So we do not, it depends on you. Then the number of patients, yes. Remember the protocol, the guidelines, there are basic number. It could be, if it's very few, we tell you that it's very few. Remember that from a particular phase, there is a limit. You have a range of participants that should participate at that level. If it's phase 2, that is very small. Then phase 3 has a level. Then when you say phase 4 is post-marketing. Yes. So then finally is whether Nigerian government gives support. No. Nigerian government as of today does not give any support for clinical trial in Nigeria. Okay.
Thank you.
Yeah, I just wanted to make a clarification here, because the term phase 4 is actually used variably by different people, and it's important to know in what context you're using that term. These days even you have what they call phase 4 commitments. So, what is called a phase 4 trial may be a trial that the regulatory agency demands to be done because of some type of early drug approval, review process that has been based maybe on surrogate markers. And I think that is what Dr. Maduke was alluding to, as opposed to, the phase 4 trials that are done for the license indication, where the objective there is effectiveness of the use of the drug under normal conditions, in the marketplace. But I think the key question here is, before you start to talk about duration of the study and the number of patients that need to be sampled for that study, the first thing is, what's the question that the study is supposed to address? And that should dictate the design. Even in Europe, you have what they call post-authorization safety studies, which clearly address safety studies with safety objectives as a primary objective. So I think the first thing is try and understand what is the question that the study is supposed to address, and then the design will be obvious, rather than whether it's phase 4 or this or that, because there's a lot of confusion out there, even in the West.
My name is Fumi Ajayi, Procter & Gamble. I have a few comments, and the first one is that I sincerely appreciate the presentation from both the FDA and also the NAFDAC. A lot of insights gained. If I go back to the discussion we had this morning, there are two areas that I see that was raised with regards to the need by NAFDAC. And I think, please correct me, one is current review efforts, and the other one I think is the future, if I just bucket them in those two. And I think one of the things that you raised is that you have some workshops that you currently put in place, especially, for example, the one in April that you alluded to. My question to you is, as we're looking into that and partnering with NAPPSA to help advance what you're doing over there, are you, NAFDAC, open to having experts among NAPPSA and people that NAPPSA can actually partner with, who are currently outside of the country of Nigeria, partner with you to develop, for example, you talk about the review of the current regulatory documents, development of guidance documents, which could include the review process. What needs to be in that review? How can the current staff be trained to know what to look for, let's say when they review bioequivalent studies or even the protocols and the other stuff? So I have the regulation as a whole, then the review process and the guidelines with the clinical trial protocol and the other stuff. And the third one that I think you're talking about is having some people help review the document as experts. Are you open to having people outside of the country partner with you along that journey, which is what NAPPSA is trying to propose?
Thank you very much for that question. Well, you know that logistics can also affect the performance of the reviewer from outside the shores of the country. We want to source experts. We also have experts from the diaspora. We have somebody who is a consultant to NAFDAC that is here. He's a member of NAPPSA. So we have them working with us, and we want to intensify the collaboration. But in the assembly of the experts, it depends on, for example, we'll assemble experts, and assuming we want to review a particular product, innovative product, on, okay, let us say the biopharmaceuticals, which is quite new, it's a new area in medicine, and we don't have the capacity to review it. We may call anybody to help us review it. For example, in the unit, we don't have a biostatistician working with us in the agency, and we have data, many things that are submitted, and we need somebody who will review it and interpret and then give us a clear comment on what is being submitted. Then it could be in oncology, it could be in any other product. So that's why we want to assemble experts. So it may be from NAPPSA, it could be from internal. But basically, as a person, I think logistics. If we, for example, want to review something and we say we need somebody from US here, and then the person will now tell us, no, I can't come this month, give me till December or January next year. And remember, the person is waiting, the applicant is waiting. So those are the things, and we may have to consider people
Yeah, I understand the issue with the logistics, but what we know that with the information highway, the electronic system, the way it is these days, that's nothing. The EMEA actually has a system where they don't have their reviewers sitting in one building. They use experts, and those are anywhere in their universities or wherever they are. Especially, the thing is they don't have any tie to the applicant. That's one of the things. So they review that, they send their comments back, and they, that's, this day and age, teleconference, you don't have to be there in person. If people are willing, the first thing is, do we have a list of the people who are willing and where they are? Are they here in the US? Are they in the UK? Are you tapping into some of the people? I was at the FT for over 10 years before I left about 9 years ago. And you have Imo here, you have several other people here, that we more or less cry over the fact that we don't have people that can look at the data. I think we will do some justice to the situation here if we know where we can serve and we are allowed to even try. If there's no biostatistician, for example, like you said, to look at the data, somebody will have to make a call, and that call will be, like you said, is a toss of the coin up, head or tail. Do I put something? Then having somebody have a delay of a week or two, actually makes more sense than have people get a product that may do harm to them. I'm just appealing to NAFTA to take a look at what they have, because what you're hearing is that you have quite a lot of us here that I can help look at the regulation, work with you, and also help develop what people need to look for in an application to actually make the good call, and also train them. You have workshop, you have people here that actually partner with you with the GCP, but that's just an aspect of it. You can do your study, you can have the data in, the data may be telling you something and the conclusion may be wrong because the applicant wants to get the thing approved. Of course, we can go on and on, but the appeal is, let's start with getting the names of the people who are interested in partnering with you. And I'm sure most of the people here are not asking for a fee, for them to be paid to help you. I'm not, I'm sure, most of the people here would like to do that rather than have their people, have a lot of products that are not, adequately vetted, given to them. And I know that you are in a position to help do this. You are interested, and I'm just appealing to you because I know you have a lot of people back home to send this idea to. So it's not you I'm talking to with regards to the obstacle, but we have you here, and we'd like to have you be our mouthpiece and our, eyes and ears when you get back to say we have a lot of people that are willing to donate their time and their knowledge to advancing what NAFDAC is doing.
In future, education, for how in Africa in general, how are we going to provide this oversight for the system that will deliver truly innovative products to our populations.
Aim at having a common regulatory framework for clinical trial regulation in Africa. That is basically the aim. So everything that goes that is needed, that is harmonization, to harmonize the regulatory apparatus that can be used in Africa, registration and regulation of vaccines, clinical trial of vaccines.
That malaria, masquerades is around the corner. Maybe in about 3 years' time, we're going to have a vaccine that can reduce, mortality in our children by 50%. It makes no sense that each African country will now request for dossier submissions and stuff like that. And the same thing even applies to post-marketing surveillance systems, like the WHO, pharmacovigilance system really doesn't work in Africa because hardly anything goes there. And you find that manufacturers will have to establish on their own sentinel pharmacovigilance systems to support these innovative products that will be delivered, to the marketplace hopefully within the next 5 years. But I think this is an area where, NAPPSA and the other regulatory authorities in Africa, need to come together and actually ask this question: do we need maybe a West African, regulatory authority, or maybe an African or sub-Saharan regulatory authority? And then it's a centralized review process, just like we have, in Europe.
I'm a clinical research professional for about 15 years. And just to follow up on the last question here, first thing is, how does an organization or individual come on the radar screen of NAFDAC? Just to simplify the question. Number two is, what is...
Sorry, I didn't get your first question.
How does an individual or organization get on the radar screen for NAFDAC, and to assist NAFDAC in some of the regulatory practices, in some of how to review the documentations and applications? The second question is, what is NAFDAC's current position regarding medical devices? There has been a lot of talk regarding drugs and biologics, but I haven't heard anything yet regarding medical devices. Because the process in the U.S. is significantly different than it is for medical devices in comparison to drugs. And so I was wondering, are there distinctions between those practices when dealing with NAFDAC? Number three is, in the U.S., there's a portal system, clinicaltrial.gov, and the last time I looked, there were about 20 to 25 studies being conducted in Nigeria. How reliable is that information? And what is NAFDAC doing to ensure that all clinical studies that are being conducted in Nigeria are actually being reported either in clinicaltrial.gov or in a similar portal that is distinctive to Nigeria? And the fourth question is regarding the pricing, because I'm a strong believer in promotion for clinical drug trials in Nigeria. However, one of the concerns I have is that, yes, we're putting our people in place in terms of being guinea pigs for these clinical studies. What some of the Latin American countries are doing, mostly coming from the regulatory aspect, is that they're ensuring that if a pharmaceutical company is interested in conducting clinical trials, that if those drugs ever get approved, that the people that participated in those drugs are not at a disadvantage in terms of pricing those drugs. A number of HIV drugs are priced at an amount that most Africans and most Latin Americans cannot afford. What is NAPPSA doing to ensure that if we do clinical studies there, that the pricing will be reasonable for people who participated in the study?
On pricing, we do not have on pricing. For the HIV antiretroviral drugs, the price from Asia, from India is 2% of the price before, and the price is coming from America and Europe. That is the truth. And then what we do is we ensure that we inspect the factories, and that's where bioequivalent studies come into play. We don't have any input, we don't have any price control. So we can't influence companies in America to change their prices because we want the drugs in Nigeria or Africa.
Can I just elaborate on that?
Yes.
Even though we don't currently have input in it, I think it's in our best interest to have input in it. Once again, with Nigeria's population, I think we're in a good position to influence. We're in a good position to influence those pharmaceutical companies.
Sorry, you cannot. You cannot, because it's a free world. The company, you can only negotiate. You can only negotiate. And remember that we don't buy drugs except the government decides to buy bulk products, especially for some diseases like and for drugs for anti-malaria drugs, anti-tuberculosis drugs, and antiretroviral drugs. Because these are the diseases that are of interest now, of health priorities in Nigeria. So like I told you, there are some companies that were making antiretroviral drugs for the Nigerian government, and they had to crash down the price because we could not afford the price of drugs from America and Europe. Because I remember then, to treat a patient for a month in Nigeria with antiretroviral drugs, was costing 60,000 naira, and no one can afford that. But eventually, when the federal government intervened and went to India, the thing was crashed down to 3,000 naira every month for the patient. And what was the percentage? Is it 5%? You understand? So that is the only thing we can do, to source drugs where we get good, effective drugs, drugs that are efficacious, drugs that will deliver what we actually want it to deliver, and at a very good, affordable price. I think that is what we owe the masses, and that's if we can negotiate. But if you negotiate and you don't succeed, what do? You cannot influence a foreign company to crash down their price because of you. That all the clinical trials that are going on in Nigeria are documented. Remember in my presentation I said, yes, some people, because of ignorance, they do not know the role NAPPSA plays in regulating clinical trials, and range from the ordinary tongue depressor, hand gloves, to the exotic ones like implants, etc. And for such exotic ones, we do not have the capacity to evaluate. Because some of them, you need to evaluate the influence of radio frequency, electronic magnetic waves on some devices. We don't have that capacity to evaluate. We depend on documents. And for sometimes they bring in those things without even us knowing. Yes, that is the truth. We are all Nigerians. We are talking about the problems we have in our country. Some exotic devices are brought into the country without NAPPSA knowing. That is the truth. And that some scanners and all that. So those are the things. So we have limitations, and we are trying to see what we can do about all those things.
Actually, I'll make it very brief. I want to thank the presenters for a wonderful dedicated presentation. My question goes to Dr. Eprobati. You did mention that in vitro tests should not be used to compare formulation equivalent to the RLP, particularly for topical dermatological products. I agree. And also, you did mention that for in vitro testing, I think rheology and the diffusion could be potential tools to use. However, from my experience, as these sometimes don't correlate to what you see in vivo, particularly they may not have any relevance to the clinical endpoint. So my question, even when sometimes we try to use cadaver skin study, I think you may find out at the end that both different formulations here, or test formulations you're looking at, reside in the same area within the skin. My question to you is that how do you select your lead formulation for clinical trial? What in vitro models can be useful in properly selecting a formulation that will help you to establish the listed and correlate to the aesthetic manipulation?
Those particular studies using cadaver skin. We do not accept any type of studies using any type of animal skin or any dead human skin. The only in vitro tests that we accept are the ones that's also on the SUPAC guidelines. Normally, for a drug product to be approved, the initial stages, the firm is going to have to conduct clinical trial studies. That's for new drug applications. So there's no way around that, really. We're going to have to conduct a clinical trial study. For bioequivalent, the studies are generally, it depends on the product as to whether it's a clinical trial or just a trial in human subjects. It's really dependent on the type of product. For the clinical trials, we're not going to take in vitro studies prior to the clinical trials. It's going to be a recommendation for you to actually conduct the trials themselves. So that's all I can say about that right now.
One question related to a previous comment and question by Mr. Fatiga here concerning compensation of clinical trial subjects in Nigeria. Some of us in the bioethics community here have struggled with how trial subjects in developing places like Nigeria can not be used as guinea pigs for trials without deriving benefit from the trials after the trials are over. So many thoughts and ideas are bandied around. And I was very pleased to hear about what you said the Nigerian government did with regards to retroviral drugs in Nigeria, which make the company reduce the price of its drug for the general population. But there are hypertensive trials, there are oncology trials, there are so many other trials, I'm sure, that are either going on or will be going on in Nigeria. I know NAPPSA is not involved or it's not set up to go into pricing or specifically concerned with ethical considerations. But is there any way NAPPSA and other organizations like that can influence the Nigerian government to ensure that some mechanism is in place to see that subjects of clinical trials are not abandoned after the trials are over?
Country where there's so much poverty, it has also disadvantage if you make money. If money is part of it.
Compensation doesn't have to be money.
Oh yeah, well, if the trial, something goes wrong with the trial, or if for the normal trial, compensation should be in the part of the protocol, is it?
Okay, let me be more specific. What you're referring to occurs everywhere in terms of clinical trial subjects in the course of the trial, in which, particularly in phase 1 trials, there are many healthy young people in this country who labor on clinical trials. But what I'm trying to say is, when a phase 3 trial is over and the company has left Nigeria, got approval for its product, to sell its product, and it's a positive result, let's say it's a hypertensive drug trial. Now, the people who were part of that study, can that company be to supply the
for now,
drug?
there's no provision for that.
Yeah,
But we look
Is
at
there a reasonable
insurance. You know, what we incorporate is insurance for the trial subjects to be insured. But for now, there's no provision that by the time you complete the clinical trial and then your product is in the market and you are keeping your money, then you can give compensation or, you know, there's no provision for that for now.
Well, isn't something too far for us to think about?
Well, that could be looked into.
Thank you.
And company, I want to join everyone in commending the speakers. I think it was very informative, and particularly I'm delighted to hear Mrs. Madikwe talk about the workshop that we had in Abuja in April and what has happened after that, which is really remarkable. That it generated so much interest, and not just the interest, but you actually took it a step way higher than we thought you would be by this time, following the workshop. So I want to commend you and the Director General quite a bit for that. And also to let you know that you have a commitment to continue to work with you to make sure that you certainly achieve where you want to be. And that actually leads me to the point that Bolahan made about, and Fumi made about access to expertise in the diaspora. And all I can say is that I think the agency is very open, very willing, and ready to work with anyone that has the expertise that they need. You just have to find a way to maintain that relationship with the agency. And what we are doing that, as far as I'm concerned, is I'm glad that you're here at this conference. Become a member of NAPPSA. Yeah, become a member of it. If you become a member of NAPPSA, you know all that we are doing and all that is going on, and you'll certainly be able to contribute your quota to helping back home. And so I hope I have motivated you enough to not only be at this conference, but after this sign up as a member and participate very actively in activities. We'll really be glad to have you, given the expertise that you bring. I don't have a question as such, but I do have a couple of, maybe 3 other comments that I just did like to touch on. And 2 of them actually relate to, or one of them relates to what Wale has just said, and the other one relates to what Balahan also said. And that is number 1, trial disclosure in registries. And number 2 is post-trial access, which is what Wale was really alluding to, in the sense that at the end of clinical trials by multinational companies, do they just pack their things and get the data and get out of the country without making any provisions for that population, I should say, or that community that granted them access to collect that data? And I want to stand here not only as somebody who has interest in making sure that my people back home receive the best care, they receive the benefit of contributing to data that is generated. But I also stand here to say that I work in a company that is very ethical, and that is what most multinational, in fact I want to say all multinational companies, certainly those ones in this country, the US, and I want to say the same thing in Western Europe, it's the same thing in Australia and New Zealand and so on, even Japan. That is what they do. If you have had a chance to review the 2008 Declaration of Helsinki, you will see that principle number 14 talks about post-trial access. Principle number 33 talks about post-trial access. Principle number 19 talks about the need for you to register your trial in a public registry before you enroll the first patient. So now, the other point in all this is that industry has significant interest in this issue, in doing things properly. Times past 15, 20 years ago that people could just do anything. It is a different environment now. And every single company has the interest of making sure that they behave. They make sure that, in fact, their social responsibility policies internally allow them to do that. Let me tell you two things. There is an access to medicine index that every company is run on these days. My company, I believe this year was number 3 or number 2. Novartis was like number 3 or 4 or something like that. Every company gets run, and that plays a role ultimately in who invests in your company. That's number one. The second thing is that there is also another component of this, which again is still a way of ranking your corporate responsibility to the community that you are doing business in. And that also plays ultimately into who invests in your company. So those are two things I want to make, that we have to be extremely careful. People are suggesting to find a way of controlling price. You have to come to a certain balance. Industry does make a lot of provision, at least my company and many like mine, do make provisions in terms of what to do with trial patients after the trial is finished. And that provision always is to think about the good of those patients and that community that they are for. Because nobody wants his name to appear or the company's name to appear on Wall Street Journal, on the Washington Post, or any of these articles on CNN. It's very bad. What it does is it wipes out half the company in 2 seconds. So we are very, very careful about that. Now let me also say this. Because just last week, I was a lead author on a paper published online, early online publication, on an article called Ethical Issues in Industry-Sponsored Trial. I lead a group in pharma, and as a result of my role in pharma is the U.S. Trade Association that is an umbrella organization for U.S. pharmaceutical companies. As a result of my role as a co-chair of that group, I co-authored, I led the authoring of a paper that is published online in Pharmaceutical Statistics.
Pharmacist, stroke pharmacologist for the UNTH at Enugu while I was with the College of Medicine. It's based on that that I want to make some quick observations as to what NAFDAC needs to be thinking about. I can see Dr. Madukwe, and I'm looking at the huge prospects of all the things you have to do for clinical trials. And it's not quite completely clear in my mind, but I want you to think of the model in Malaysia, at the University of Science in Malaysia. They have a huge clinical trials unit where people from the so-called developing countries can actually send their staff to actually train by WHO. And it's a model that's really great. I mean, they can teach you all kinds of things. Because I see a lot of need for capacity building in your unit. And as I think more into clinical trials, I begin to understand that it can be overwhelming, especially because you're still dealing with the basics of drugs that are fake. And right now, that's a lot of effort on your part, draining a lot of your staff that you don't have already. But anyway, the points I want to make is, I was a clinical monitor as the only real clinical pharmacist at UNTH for the artemisinin trials that came to UNTH and the halofantrine trials that came to UNTH. Dr. Amina and myself and a few other physicians were there. And I know there was some representation from the Federal Ministry of Health. But my concern as a clinical pharmacist is that some of the serious adverse events we actually noticed and picked up happen to have been kind of put under the carpet, to the point that halofantrine, for instance, some of our clients were beginning to have some neuropsychiatric side effects. Okay, one man, for instance, kept talking about his standing from his refrigerator and falling down. And of course, here we are. This is phase 3. Okay, and nobody has told us from the company side if they actually experienced all that. So what I'm saying is, you also need training at the level of the teaching hospitals and need to be sure that things are there for them to really do the testing. Some of the EKG testings we did for out of country, some of the things we did for atomicinin. The centers that are chosen have to be centers that are able to do that and have seasoned people, hopefully a well-trained clinical pharmacist, along with the physicians to do all of these, to look at the drug-drug interactions, all that. As you know, I published a lot of ivermectin. The original ivermectin studies that established ivermectin as something that's safe in pregnancy was really my idea. It was my work, and I did it for like almost 3 years before I came back to the States. So drugs in pregnancy is another concern. You know, there are many women who get into these trials and maybe they're pregnant. What are the categories of the drugs there? You need input from all kinds of people. You need clinical pharmacists, you need oncologists, you need people in regulation to really get at it. And sometimes I wish there was a body that's sitting there for you to use. But like I was saying, you have the expertise in this room, and you should feel free to tap on it. I want you to be aware that between 1999 and 2002, a total of no less than 400 drugs were withdrawn from the market, mostly because of problems like Tosades, the point, arrhythmias, problems that were not reported in their clinical trials, but were actually seen on the field. If you look at all the problems we're having with suicidality on the SSRIs and the antipsychotics, some of which Pfizer had hidden, those are things that we have to keep watching out for our people. Because, you know, as you see, I don't mean to cast any aspersion on anybody, but we have to be careful and make sure we're not a dumping ground and people come and do trials without telling us what has gone before.
In Virginia, I've been involved in clinical and non-clinical drug development for about 13 years. Prior to that, I was at the University of Georgia. Anyway, my first question is, from the time you submit a clinical trial application, from the time I walk into NAPPSA office, to the time you give me approval to start the trial, how long does that take? That's one. Two, if the drug I want to try or I want to conduct clinical trials in Nigeria has never been approved in Nigeria before, how many trials will you require me to conduct? If it has been approved somewhere else in the world, not Nigeria, how many trials would you want me to do?
So I think it's that.
Okay. If it's been approved, if it's never been approved, you've been facing with the conducting clinical trials in Nigeria, that investigators and clinical trial staff are not very familiar with GCP. And I think that is an issue that should not be taken lightly. My recommendation would be that there's an association of GCP of Nigeria.
Yes.
Why don't you work with them?
You are working with them.
To get them to set up a program where they can certify sites and investigators, and if they don't take the course and get a certificate, don't approve the site for a trial.
When you finished your trials and then you submit the report, isn't it? How long does it take for us to review it?
No.
How long does it
If I
take?
want to start a trial,
Yes.
how many, from the day I come to you to say, I want to conduct a clinical trial, I fill out the papers,
Yes.
how long will it take for you to give me approval to say, go ahead and start the trial?
Well, if, for example, first and foremost, remember that you have to come and get the guidelines. We give you the guideline. That is this new guideline that we developed now. So with that, actually, it's like we've given you the format, everything on how to go about preparing your protocols. Okay? Now, if you do it rightly, and we are satisfied that everything, your study design, everything, all the things that you must put in place are there. Within 2 weeks, we ask you, we approve. Even within 1 week, we approve for you to go and start your study. Now, for you to conduct your studies, now it's your own you know when you complete your studies. We don't have any listing.
Okay, do you require me to submit the report of my ethics board review?
Yes.
Okay.
Now you have to show us evidence that the institutional review board, that you have approval from NREC.
Okay. All right. The other question will be, as part of that, when I finish my clinical trial,
Yes.
and I have a clinical trial report,
Yes.
and I submit it to you,
Yes.
how long will it take you to say, go ahead, you've been given approval to market this product?
1 month.
Thank you. And then when you have
Now, then the other one is, there was another question you asked that if a clinical trial is not approved, sorry? There was a question you said,
okay, I said if the drug has never been approved.
has never been approved
Anywhere
anywhere.
in the
It's a
world.
fresh trial.
Yeah, it's a brand new drug,
Yes.
new chemical entity or whatever,
Yes.
new
Now,
herbal.
For that, you do it in 3 sites, geographically spread in Nigeria.
3 sites.
Yes, 3 sites.
Yeah, but it doesn't mean 3 trials. You mean 1 trial, 3 sites.
That's multiple sites, but yes, and the design will be the same thing.
Yeah. You say, okay, the reason I'm asking you this, if you go to some regions of the world, like here in the United States, or in Canada, or in Europe, they require you at the minimum to submit 2 clinical trials. So it will be the same design. That's why
We can approve 2, but most times we ask for 3.
No. 3 sites
Mm.
does not equate to 3 clinical trials.
No, I'm saying 3 sites. For example, if you want a particular clinical trial, having this for the same product,
Yeah.
same design, everything, you have 2 or 3 sites.
Included in the trial, in the one trial.
Yes. Geographically spread.
Okay, thank you.
Now,
I think I understand.
and then if the thing has been tried in another country before, now you bring the report to us.
Okay.
We look at that report. If you did not carry out that trial, there is no representation of a black community. That is, some of the trial subjects are not black. And even if that you must have the number that is, that will accept. Okay. Yes, yeah, assuming you do it in phase 3. Assuming you do it here in the US, and then there is no black population, we don't accept it. We may ask you to repeat in Nigeria, but if you did it elsewhere that represents black population, we accept.
All right. I think I have an idea.
Yeah, I think what Dr. Maduke has just clarified is that, as opposed to the requirement in the West where you need 2 adequate, well-controlled trials, in the clinical indication, in Nigeria, you always need 1 trial. It seems like it's the number of sites that, may vary, depending on, the extent of, trialing in, black populations, or whether or not it's a new chemical entity that has not been licensed elsewhere. That's what I think I heard. Okay.
Thank you very much for your presentations. I just wanted to make a comment, in light of the big pharma. I know we tend to get swear words, and these days our reputation is often even lower than those of the tobacco companies. However, but I do want to raise this because this is an important issue, because as we're thinking about Nigeria growing in this area, we need to grapple with this. I think Imo had talked very clearly around how most big pharma, and I've had the opportunity to work with at least 2, and really take the ethics seriously. I know that any time there's an issue, the public outcry is always that the big pharma covered it up. The reality is that, as we all know, none of our preclinical models predicts for human beings. And as you go into trials, the rarer a serious adverse event is, the longer it takes before it shows up. So sometimes things show up in phase 3 that never showed up in phase 1 or 2, simply because the numbers were not large enough to be able to get an incident. Sometimes things don't even show up throughout development and don't show up until the drug has been in a large enough population that has used it for a long enough period. I say this because we have in Nigeria have had a history where we've had one or two very internationally publicized trials that people perceive things went wrong. Now, it certainly wasn't in that company, but I would like to believe that they meant to do good. Perhaps one or two people were not as proficient as they could have been, and attorneys, being attorneys, always like to take advantage of facts, to take advantage of things that will help them. But dial forward, if we're going to go forward, and if we want to grow in this area, one risk that we have anytime you're going to take new drugs is the risk that something new may occur, and even something that may have been tested, just as you were talking about in different populations, that may have been adequately safe, may end up coming in there and showing some safety signals that weren't seen at all. I think we need to build some transparency. We're going to be counting on NAPPSA to help maintain and ensure that transparency. We certainly don't believe that any pharmaceutical company wants to make Nigeria or any emerging market a dumping ground or a testing ground for things that are not safe anywhere else. But we also have to have legislation and regulations to ensure that those trials that come there have as best a data safety package that is possible, preclinical or clinical, depending on the stage at which it is coming, and be prepared to work with the public if an unfortunate, unexpected safety event occurs. It happens here all the time, and when it happens, we wrestle. Every drug that gets approved has most drugs have experienced more than one hurdle, and many times drugs still get approved because overall the regulators are looking at the therapeutic advantage, the efficacy and what it delivers relative to the issues. And those package inserts that are written in small font to contain all the information you need is meant to help both the physician and the patient weigh that risk. It will not be different for Nigeria. It will not be possible to be bringing completely clean drugs that have no adverse events. But we'll need to work through this, both as a regulatory body and as we educate our organization and educate those who take part in trials. This is one of those risks of being able to get new therapies for our nation and for our citizens. Thank you.
Thank you.
At that time, whether it's at the time of licensure or at any time point during the life cycle of the product, post-marketing. Now, what is the real issue about safety today? We have safety challenges.